Grab your eggs and have a seat, please. And let's just verify that osmosis is a real thing that happens, because it sure did. Pretty dramatic, right? Pretty dramatic. What's happened to your eggs? Give me the short version. What happened to your eggs?
Imploded.
Nope.
All the liquid from the inside left.
Yeah, the water left the egg. We have a shriveled up little raisin of an egg now because you put your eggs in a concentrated sugar solution. Right? It works. Even over the weekend. It works. Osmosis is real. That's kind of the point of this. So what we are going to do now, your second solution, go ahead and log in and grab get into your egg labs. I'll be right back. All right, so solution 2. The solution 1 was corn syrup. Solution 2 is distilled water. Do you know what distilled water is? Oh, gosh, first period. Brilliant. Here's the kind of Stone Age way to distill water. You have 2 pots, okay, and there's water in the one pot, and if I could draw fire, but okay, here's fire. You heat the one pot till it boils, right? And then you have an empty pot, and basically just a slanted piece of metal above the 2 pots, so that the steam, as the water boils, condenses on the metal strip and drips down into here. And what you're doing is boiling the water off of all of its solutes, if that makes sense. So what you get in pot number 2 here is distilled water. This is the process of distilling, and it's the purest water you can get fairly cheaply. So you are putting your egg now into the purest water we can get. It's just another beaker problem. So you know what's going to happen. So your hypothesis should be pretty, high confidence hypothesis. Now, as far as your procedures, this is the day I need you to be as careful as you can, because corn syrup is gross. When you dump it in the sink, please make sure there is a stream of warm water going, and that you make sure that corn syrup kind of makes it down the drain and doesn't get on the other stuff it could make sticky. It basically fossilizes and turns into this super hard crust that we have to chip off. So please be neat with your corn syrup. You're going to dump it into your hand like you did before. You're going to rinse it off, okay? And this will take a little longer. Just make sure it's a gentle stream of water. Don't you dare turn that faucet on with something under it. That's a booby trap, right? And so you rinse off the cup and egg, make sure they're, corn syrup free, and you do the weighing thing where you zero the cup, and then you weigh the egg in the cup after you've zeroed it to get the mass. Then you'll calculate, of course, the amount of mass that was lost between, pre-solution 1 and post-solution 1, and then you use that mass also for the Okay, so the eggs are in their distilled water. I wanted to make sure that you saw this. I think I forgot to click on the last part of this slide. We had hypertonic, where water leaves the cell, hypotonic, where it enters the cell, and then there's isotonic, which is equal concentrations inside and outside of the water, goes in and out equally. Am I right that I missed that? Yeah, just go ahead and fill that in. And for your assignment tonight, that'll be necessary. So for the last bit of this chapter, the question we got to answer is the question you guys always come up with. It's a great question when we're doing problems like this. The question is, Sandro, if that stuff's too big to cross the membrane, how did it get in there in the first place, right? So that's the question we answer today. Because you got to get stuff like that in and out. Because, glucose is something the cell needs, right? There's various wastes that have to get out, and they're too big to cross. And so there's two answers to that question. Answer number one is called active transport. Active transport is a kind of transport that requires energy. But because it uses energy, it can move solutes, big ones that are too big to cross normally. It can move them into or out of a cell against the laws of diffusion. In other words, it can go from low to high concentration if you want to. You can pack something inside or outside the cell, no problem if you're spending energy to do it. Now, there's a couple of examples I'm going to give you of active transport. The first one you've already seen, and that is endocytosis and exocytosis. I'll remind you that when a cell, like an amoeba, or like one of your white blood cells, finds something that it wants to get inside it, one of the ways it can do that is by yanking on its microfilament network and changing the shape of the cell and bending that cell membrane around the thing that it wants inside it. Does that ring a bell from the endosymbiotic theory? Yeah, so like that is a process that it's called endocytosis that lots of cells do to get stuff inside them. The opposite, where you take something you want to get rid of, you wrap a membrane around it, and then you send it to the cell membrane, fuse it with the cell membrane, open up the cell membrane, and spit it out, is called exocytosis. That's it right there, exocytosis. I would recommend drawing at least some version of one of those so that you remember. Again, it costs energy. But you can put stuff where you want it. Questions? There's another example of active transport. Very specific example. Oh, that's probably a good thing to say, too. That's how the amoeba eats and how some white blood cells kill bad guys. So there's one way you can get stuff into or out of the cell that might be too big to cross otherwise, using energy to do it. Way number two is called the sodium-potassium pump. Now, there's other kinds of pumps as well, but this is by far the most common one in your body. Na and K, if you haven't learned it yet, they're good ones to know. Na is the periodic table symbol for sodium. K is the periodic table symbol for potassium. The sodium-potassium pump does kind of what it sounds like. Just about all of your cells have them. In fact, they turn out to be so important to the functioning of your muscles and nerves that you spend about 1/3 of the total energy you get from your food just on running these pumps. So important there. They use energy as it is active transport. And what they do all day is pump 3 sodium ions out for every 2 potassium ions they pump in. So if you're doing that all day, which you are, you're running your sodium potassium pumps all day, every day, pumping 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in all day long, what's that going to do? It's going to use a lot of energy. What else? What are you going to have a lot of inside the cell? You're going to have a buttload of potassium inside the cell. You do. What are you going to have outside the cell?
Sodium.
A buttload of sodium outside the cell. That's sort of effect 1 and 2. The third thing is a little less obvious. If you're pumping 3 positive ions, and that's just a charged version of an element, an ion, right? 3 positive ions out for every 2 positive ions in. What is the inside of the cell going to become? You think of the cell as a mix of a bunch of positive and negative ions, and suddenly you're pumping 3 positives out for every 2 you pump in. Vinny, what's going to happen? Yeah, the inside of the cell is going to become slightly negative, along with the high potassium concentration inside and the high sodium concentration outside. We have a negative charge in that cell. Those of you that go further in your biology education will find out that that pump is basically the whole setup for the function of your nerves and muscles. That negative charge sets up an electrical potential. That means that your nerve cells can work basically like electrically conducting wires. It's so cool. But it's a little too much for us right now. Here's what it looks like. This is the pump. This is the potassium floating in. This is the inside of the cell. This is the outside of the cell. Here come the 3 sodiums from inside the cell. They lock into these 3 spots. This is a phosphate from ATP, which we'll talk about, that runs the pump once, letting the sodium out, and the potassium floats in. And all day long, that's what your sodium potassium pumps are doing, setting up the electrical potential that makes your nerve and muscle cells work. So, endocytosis, exocytosis, and the sodium potassium pump are our 2 examples of active transport. Then there's a type of passive transport called facilitated diffusion. Facilitated diffusion works because of channel proteins, which you've seen before early on when we talked about the 3 kinds of membrane proteins. Remember we said one of them was transport proteins? Well, the ones you saw were channel proteins. Channel proteins are just proteins with holes in them, basically. Very specifically shaped holes that let certain things go through the membrane, as if they were small enough to do that normally. This doesn't cost any energy. Really, the word facilitate just means to help. Like if I facilitate a meeting, I'm not one of the interested parties in the meeting. I'm just there to help make sure the meeting goes slowly. So this is basically diffusion with help, with the help of a specifically shaped hole to diffuse through. It doesn't cost any energy. It's very specific, but It is restricted to the laws of diffusion. Can't get anything to diffuse in or out using facilitated diffusion against the laws of diffusion. In other words, it has to be high to low concentration. If you want sugar to diffuse in through the glucose channels, you better put more concentrated sugar outside the cell than there is inside. And remember these three kinds of membrane proteins. This is the marker protein that shows your body what kind of cell this is. This is a receptor protein like the ones you use in your sense of smell. This is that channel protein that allows facilitated diffusion to happen. So active transport costs energy and can go against the laws of diffusion if you want it to. Facilitated diffusion is free, except for the energy you use to build the channel proteins, but it's not going to go against the laws of diffusion. Questions? Riley?
This might be useful. Is this a different color being like highlighted or something, or just different color?
The green type, you mean? I don't know why it's green.
Some just do it.
No, I just apparently was scaling green. Good. So that is basically it for the chapter. You're looking at a test on Wednesday. What I'd like you to do for tonight, if you'll get out your second set of beaker problems real quick. The ones I gave you Friday that I said would be a real grade. We're going to do the thing like we did with the other cell assignments, where you're going to get to put your answers into a Google form. Consider them. Make sure you're understanding solution types, because those are in there too. So I'm going to drop this form in. I'm going to do tomorrow. And basically, if you're confident in your answers to those beaker problems, you're just transferring those answers in. But you have a chance, if you're not confident, to kind of rethink them, knowing this is for a grade, knowing that the options that are there are the ones you have to choose from, and go ahead and complete that in the best way that you can. Any questions about that? You'll notice, and this is important, there's no solution type asked for when there's no water moving, which is correct, okay? Hyper and hypo and isotonic only exist when something is stuck. If it's all, CO2 and oxygen, we don't worry about solution. Water's not moving. Caleb, you had a question.